So I'll start. Hi everyone, my name is Sally and I'm a PhD student from Hanadi Sleeman's lab at McGill University. And I happen to be here to share my project on enhancing mRNA stability through small molecule nucleic acid supramolecular sampling. So as you all know, the bottlenecks of the field of nucleic acid therapeutics is that nucleic acid suffers from low stability. And also there are delivery problems such as low endosomal escape, biodistribution, and non-specific uptake. And for this talk, I'll mainly focus on tackling the low stability nuclease resistant problem. So just to touch on the structure of the mRNA. So mRNA has a 5' cap and 3' poly A tails and two non-translating regions at 5' and 3', and also the coding regions of your protein of interest right in the middle. And why is it very hard to protect mRNA? Because first of all, mRNA consists of 100 to 2,000 of bases of nucleotides. And also, the second reason is that from its innate structure, the 2' hydroxyl group can accelerate intramolecular cleavages. So at any point of the 1,000 bases, any degradations will lead to an inefficient and insufficient translation. And there are already strategies to increase the mRNA stability, but they are all mostly sequence-based. So first, you can have modification on the 5' cap, or you can use computational tools to maximize the folding and the structure of the coding regions. And also, you can use a sequence that is known to recruit RNA binding proteins, or you can even lengthen the poly A tails. And really well-known method is also to use chemical modifications. And for mRNA, it's synthesized enzymatically. So mostly we have modifications on the nucleobases. However, the limitation for this method is that first, it requires a complex synthesis for the nucleotide triphosphate. And also because mRNA is synthesized enzymatically, every modification you want to incorporate, we have to test in the incorporation efficiency, the scalability, and then enzyme fidelity. So all the methods I introduced here has a common theme where we substitute a component in mRNA, or we put mRNA into a geometry that's not recognized by nucleases. And all of them is sequence-based, so it's laborious. So here this is where our lab proposed a new methods through where our Sleeman lab study DNA, and we see DNA as a material. We study its supramolecular assembly. For my project, I'm mostly focused on a small molecule called cyanuric acid. And what is interesting about this small molecule is that it's a nucleobase-mimicking small molecule that has 3 thymine phases. And it's highly symmetrical. So what it does is that not only binds to the Franklin-Watson-Crick phases of adenine, like thymine, it also binds to the Husting phases of adenines. And with the presence of cyanuric acid and adenines, it will form this continuous hydrogen bonding network, shown here. And from this animation, you can see that cyanuric acid can act as a hydrogen bonding glue that take together 3 strands of polyadenine together to form this triple helix structure. And later on in this talk, I'll refer to this structure as triple helix or as fibers. And what this research entails is that it expands the DNA codes with minimal synthetic complexity, and also cyanuric acid small molecule itself and the structure is non-toxic. So previous member from our lab, Dr. Lachance, used this structure as a material to develop a DNA hydrogels. So what he did is that he not only had polyadenines, but also have the cross-linking regions that encodes for I motif or for duplex or for quadruplex. And with the presence of cyanuric acids, he was able to form the stiffest DNA hydrogels on records. And what is really interesting about this material is that it has stimuli responsiveness to pH, to strand displacements, or small molecule comparator melody. So you can assemble and disassemble the hydrogels as you want. And this is where I joined the lab, where I was curious to see that this non-canonical triple helix structure can have any resistance against nucleases. So we started with just single-strand polyadenine sequences, and we observed that it's quickly degraded by nucleases, which is expected. However, when we add cyanuric acid inside, forming this triple helix structure, we don't see any degradation over the course of a month. And this makes us ask the question, what about a cargo sequence, which is next to polyadenine strands? as cargo sequences usually encodes for therapeutics. And what we see here is because cargo sequences doesn't have consecutive A's, it won't be incorporated into the fiber, but will be protruding out of the triple helix structures. We see a partial protection with a half-life of 1 to 2 days. So it makes us ask two questions. The first one is, what's the correlation between the poly-A and cyanuric acid fiber formation and nuclease resistance? And also is what's the extent of cargo sequences we can protect? So as we start with designing a series of strands called regiometric strands, where we play with the ratio between the polyadenine strands and the cargo sequences. All the strands shown here has the same length of poly-A's, but with different length, with increasing lengths of cargo sequences. And we did a quick serum stability test where without cyanuric acid, everything degrades within a day, which is expected. However, for the one that has cyanuric acid, only the DNA 1-to-1 strands show a significant difference when the cyanuric acid is added. However, for the DNA 1-to-3 strand that has the longest cargo sequences, we don't see any difference between with or without cyanuric acid. And then we did a direct characterization of the fiber formation through circular dichroism, where if we have a triple helix structure formed, we will be able to see a 252 nanometer peaks. And here is consistent to what we see from the serum stability assay, where from CD, we see that the DNA 1-to-1 strands that has a significant protection when we add cyanuric acid forms the most fiber, and the one that doesn't show any differences, the DNA 1-to-3 strand, shows minimal fiber formations. So this is the first piece of evidence just to show us that the correlation between nuclease resistance and fiber formation, and also it tells us that the greater proportion of the cargo sequences in comparison to poly-A sequences, there will be less fiber forms and there will be less nuclease resistance. And then, motivated from this, we want to develop toolkits that push the fiber formations. So the first thing that came in mind is just to add fuel sequences to help the triple helix formation. And by fuel sequences, they are just free polyadenine strands that add into the sequence in the presence of cargo sequences. And what we hypothesize is that if we have polyadenine strands, the fuel sequences in the system, you can space out the cargo sequences shown in blue. So if the cargo sequences are too close together, it will have the steric interference, but if you add the fuel sequences, it will space it out, helps the fiber formation. So it is exactly what we see here. So if in the presence of cyanuric acid, when we add fuel sequences, we see that especially for DNA 1-to-1 strands, we are able to see the bands even after 5 days, which gives us a 6-time increase from the one that doesn't have any protections. And even for the DNA 1-to-3 strands that didn't have any difference between with or without cyanuric acid, with the presence of fuel sequences, we can push the fiber formation and then better protect it and see the bands even after 3 days. So then, other than the first toolkits of adding fuel sequences, there's another parameter we can play with our system, which is the modifications on cyanuric acid, the small molecule itself. So the work from our colleague, Dr. Lachance, what he did is that he discovered that only 2 phases of cyanuric acid is needed for this triple helix structure formation. So it gives us one more degree of freedom to play with the side chains of the cyanuric acid. And we have a series of positively charged amino side chains with different lengths. And when we see the fiber formation from atomic force microscopy, from the C2 to C4 strands, which has shorter side chains, we see a planar fiber growth. So the thickness is around 5 nanometers. Once we push it to C5 or even C6, we'll see this interfibril stacking within the triple helix structure itself. So starting from 5 nanometers, we can push all the way to 50 nanometers in thickness, imagining those fibers lay top of each other, making this fiber swell. So this is where we ask the question to see whether the interfibril interaction can affect ultimate nuclease resistance, and how can we utilize this to protect our nucleic acid. So what we see here is for DNA 1-to-1 strand, when we just have cyanuric acid, we can still see the band after 48 hours, but mostly it's degraded because we form this structure, form the triple helix structure with our DNA 1-to-1 strands, and all the cargo sequences are still exposed outside of the triple helix structure. However, if we push it all the way to C6, to C5 to C6, where we can undo this interfibril stacking, we can effectively hide the cargo sequences in between fibers, which limits the access of nucleases to degrade them. So this is the first evidence just to show us that with the simple side chain chemistry of the cyanuric acid, but no modification on the nucleic acid, we'll be able to protect the nucleic acid and inhibit the nuclease axis. So now with the toolkits we have, which is the chemical modification on cyanuric acid and also by adding phylo sequences, it really drives us to want to investigate whether we can protect mRNA, which natively have a significant polyadenine structures. So this is my project where I want to implement this polyaCA structure to mRNA to increase its nucleus resistance. So just to go through the assay, the serum stability assay that I'll be doing, I'll talk about is we have two sets of buffer, one with or without cyanuric acid, and you put your nucleic acid of interest inside. I slightly anneal it, add the nucleases, and take time points to run gels to see whether the nucleic acid degraded or not. So the first question that might pop into your mind is that if mRNA itself has a significant polyadenine structures, why don't we just add cyanuric acid and it will form this triple helix structure? So this is the first thing we actually see, where we use the direct characterization, the circular diacharism, and you'll see here for the sample that just has mRNA plus cyanuric acid or plus one of its cyanuric acid derivative, you don't see any difference in the signals on circular diacharism. And if we have a triple helix structure, we should see a big peak at 255 nanometers. This indicates that there's no triple helix structure observed, so the poly-A of the mRNA itself wasn't sufficient to make this polyaCA fiber on its own. So it motivates us to use one of our toolkits, which is to use the phylo sequences to drive the fiber formations. And we started with the 3' exonucleases because the poly-A's of mRNA is at the 3'. So we hypothesized that if we add in phylo sequences, and then it incorporates the poly-A of the mRNA, we should be able to protect it against 3' exonucleases. So what we see here is we tested with two exonucleases. One is RNase R and one is XOTs. So RNase R just cleave much faster than XOTs. What we see here is without cyanuric acid, both of them degrades. For RNase Rs, it degrades within minutes. And for our XOTs, you can see that after 30 minutes, then the band starts smearing. But however, when we add cyanuric acid, we see that we can preserve the bands even till 1 hour. And for the XOTs, we can see the band at the same level after 4.5 hours. So we observed a 5 to 7 times increase in the half-life. And this is the first evidence showing us that the phylo sequences does facilitate incorporation of the mRNA's poly-A into the triple helix structure. And we did increase the nucleus resistance of mRNA. Then just working with exonuclease wasn't interesting enough, so we moved to fetal boron serum, which provides us not only exonucleases, but endonucleases. But it was obvious to us that if we just incorporate the poly-A's of mRNA into the fiber, all the cargo sequences, everything, the encoding region of mRNA will be exposed, and it will be very easy for endonuclease to cleave it. So it makes sense for us to not only incorporate mRNA into the fiber, but into our polyaCA hydrogels, where we envision the polyaCA hydrogels can act as a physical and steric shield for mRNA and can slow down the diffusion of the endonucleases that was initially in the supernatant. So the bottom layer is the hydrogels incorporate the mRNA, and the supernatant is the source of nucleases. So what we see here is that with the presence of, without the presence of cyanuric acid, everything degraded within minutes. However, if we incorporate our mRNA into hydrogels, we were able to see the bands even after 30 minutes, indicating that we are able to incorporate the poly-A's of mRNA into our hydrogels and can effectively slow down the nucleases access to our mRNA. And one thing we thought would be interesting to try is based on an observation done by my colleague Tyler Brown, where he observed that with phosphorylsiated sequences, when you dose enzyme nucleases, nuclease will still be able to recognize it, but wouldn't be able to bind it, to cleave it. And after all the nucleases are bind to the PS sequences, he will then dose in some PO non-modified sequences. And he was able to recuperate the PO non-modified sequences undegraded. So this inspires us to see the PS sequences as a trap for nucleases. So the way I incorporate this into my design is to first I have the bottom layer of our hydrogels. That's the same thing as the one I shown earlier, where I have my hydrogels and I put in mRNA and I incorporate the mRNA into the hydrogels. And also we have another layer, a top layer of hydrogels, where we form hydrogels, but also we have a PS overhangs. So we envision our hydrogels acting as a scaffold for the PS modifying strength to reach out. And with a supernatant having nucleases, when it diffused down to the top layer, we envision that the top layer that has a PS overhang will act like a fly trap to capture the nucleases and prevent further diffusion of the nucleases down to the bottom layer where we have the mRNAs, so we can better protect the mRNA. So I did a little screening of the top layer hydrogels where I started with no overhang, just the same composition as the bottom layer. And also I have another samples with PS overhang and then another with the PO overhang. And the PO and PS has the same sequence and same length, just different backbone chemistry. What I observed here is that only the samples with PS overhangs were able to preserve the bands after an hour in 10% fetal boron serum. And this is the first evidence showing that our poly-ACA hydrogel can act as a scaffold to set the PS overhang as a fly trap for nucleases to capture them, and we can better protect mRNA in the bottom layer of our hydrogels. And also, it's another evidence showing that PS overhang can effectively prevent further diffusion of the nucleases because they act like a fly trap. And last bits of data I have is just to show whether our nuclease-treated mRNA protected with our toolkits can be successfully translated in cells. And here the mRNA we use encodes for GFP. So same thing, we collect our sample at different time points, then we'll purify it. So we'll purify all the nucleases and leave only the mRNA inside. And we'll transfect the purified mRNA into cells. Later on, we'll read it with plate readers. So what we see here is for RNAs are the one without any protection, just mRNA itself, within 30 minutes, everything is degraded. However, if we protect it with our cyanuric acid, we'll see the fluorescent signal still preserved over 30 minutes. And same thing for 10% fetal boron serum. Without protection, everything degraded within 30 minutes. However, when we put it in our hydrogels, we're able to preserve at least half of it over 30 minutes. And this leads to the end of my talk, where I hope hopefully I convince you that our toolkit is broadly applicable to most mRNA sequences, as all mRNA natally has polyadenine strands, and it's a really easy toolkit to use just by adding cyanuric acid and mRNA together, and simply mixing it, it will make this triple helix structure. So it's very applicable and it's very easy to use. And everything here is based on a supramolecular assembly. So everything is reversible and you can tune it. You can release the mRNA whenever you want. And hopefully this toolkit can reduce dependence of the storage condition and help, which is one of the biggest challenge in the mRNA therapeutic. So I would like to acknowledge my PI, Dr. Hannati Sleeman, and my mentors and the funding agency and everyone in the lab. And I put my email here. If anyone has any question, don't hesitate to reach out to me. Thank you.
Yes, thanks a lot, Sunny, for this very clear presentation. So let's start with actually with the final part of your presentation. So you mentioned that you tested the new formulation in cells. So I kind of had the impression that these were cells in a test tube, or were these like confluent cells? Um, can you explain a little bit more about, because I think that's important, right? Because you can make a formulation where you have like a more stable component, but also it needs to do something, right? It needs to be translated. So yeah, so that's my question. So how did you test this in cells, and how to make it even more complicated and test it in more difficult systems?
The way we test it in cells is first we collect time points, nuclease treated mRNA, and purify them. So we have different time points of samples. Then we
Mhm.
purify them so the sample itself only have mRNA. Then we're not testing the delivery or anything. We just want to make sure the mRNA still work. So we just transfect everything with lipofectamine and back into the cells just to see the expression. So we wait for overnight and then we read it through Play Reader, if that clears it.
Yeah, so then my question is, because so what would be the main strength of this technology? I kind of assume that this is um, less or more nuclease resistant, right? But also,
Yes.
so how to test that these compounds are still functional?
The the mRNA compound, you mean like it's
Yeah,
functional?
yeah. This still translated into
Um, because the um you want to your question is whether our mRNA is still functional after it's treated
it
and
can still be
protected with.
translated, and because I think there must be kind of an so let's start with another question. So what would be the
Okay.
application of this strategy?
Okay. Yes. So first of all, we haven't the because we are at like the preliminary stage of the technology where this is for now, I can see it as like a preservative. For example, if we put it like in a carrier itself, you can protect it against any degradation. And all the condition we use here is we have nucleases and everything is done at 37 degrees. So I imagine this first as a preservative for mRNA when you put in like a carrier, let's say LNP or anything, and then we haven't tested its stability, or didn't have the chance to see the structure itself in cells or in human because we're not that at that stage yet, but we just for the cells work here, we just did everything, the nuclease test on bench, then we transfect everything, just the mRNA itself into cells.
Yeah. So did you? I think you also mentioned the cargo, so that the cargo has an effect on the stability. So can you elaborate a little bit more about the different kinds of cargo that you tested?
Yes. So the cargo we have here is just with various lengths. So we want to see the effect of the length of the cargo in fiber formation. So what we observe here is that everything we have here is non-modified DNA, and the fact the what we want to see here is that if we have a longer cargo, will it affect the fiber formation? And what we observed is that when you have a very long cargo, let's say the DNA 1 to 3 strands have 15 mers of A, but 45 mers of cargos, it'll be very hard for itself to form a triple helix structure. Whereas compared to DNA 1 to 1 strands with 15 A's and 15 cargos, it was easy to form the structure. And therefore, it has a better nucleus resistance. So we would just want to test the limits of the ratio between A's and non-A's to see what's the limit we can form a fiber and then to use this toolkits.
Yeah. So also, you talk a lot about so this is mainly about the length. So did you also test the composition of the cargo? Because when you have different composition or different kind of cargo, that might also have a different effect on the fiber formation.
That's a good question. So we didn't test the composition as we didn't test modification or we didn't test sequence. So all the sequence we selected was just to all non-modified. And then the way we select the sequence is just to minimize any secondary structure. So we want them to be single strand and very accessible for nucleases. And that's the component we took into consideration when we're designing the cargoes, but not the with modification or any sequence specifically.
Uh, because that actually was my follow-up question. So if you just have like because most of the cargo should form like secondary or tertiary structures. So would it also not be of interest to test these kind of cargoes?
Definitely, yes. Because the reason why we choose like single strand is very because we want it to be the most accessible to nucleases. Imagine that if you have like secondary structures or hairpin structure, it might create, at least for our purpose of study, it might create bias that tell us that okay, because it's because there's secondary structure, the nucleus is not degraded. But we want the nucleus not to degrade our strands because of our triple helix structure, not because of the secondary structure that's induced by the cargo sequences. But it's definitely worth trying out, like with different secondary structure and to see if there's affect anything.
Yeah, and then I forgot to have a look at the Q&A. But one last question for myself. So, but what about then the PS overhang? Why is there such a difference in stability?
So the motivation of using PS overhang is because the observation we had where PS overhang would kind of trap the nucleases. And when you trap the nucleases, it is occupied that it won't be able to degrade other non-modified single strands. And what we observe here is when the nucleases are trapped and we put in more non-modified PO sequences, we're able to recuperate the PO sequences and without harming them. So then it was interesting to me where I want to incorporate this into my design. I have a top layer of our hydrogels, and I have the PS acting as a fire trap protruding out of the triple helix structure. So we use our hydrogels like a scaffold, basically, and then the PS is like a fly trap. On our hydrogel, which will capture the nucleases. And what we observed is that yes, when we have the PS overhang, specifically PS, we will capture the nucleases and we were able to have a better stability for mRNA that is in the lower hydrogels.
Good, thanks. Yeah, I will quickly do the questions from the chat. I think we covered most of it, but we can maybe just quickly go over it. So, first question is, how do you envision the application potentials of the tech in mRNA therapeutics?
Yes. So for the application for this technology, first of all, the most direct thing is just to have it as a preservative added in the LNP. And we aim to, because this technology could potentially like lower the thresholds like and the requirement we need for transportation and for storage because everything for mRNA right now mostly it's stored at -20 -80 which really limits accessibility. So this is one goal we have in mind. And also if we can play with some modification on cyanuric acid, it can not only just be in the LNP, you might still preserve the structure itself in the in vivo, which can prolong its translation as therapeutics. So that's still ongoing work, but there are many things we can try because we are working with a small molecule, but not working on the mRNA itself.
One short question, and please a very short answer. Is it easy to filter off the protective reagents from the mRNA? And if yes,
Yes.
how? So
Yes.
please, a
So
short answer.
Of course. So this is everything is supramolecular. Everything is done by hydrogen bonding. So you can easily do a Amicon filter, the centrifuge filter, and you can easily screen out the small molecule.
Cool. Thank you. So I give the word back to Erin.
Great. Well, thank you, Sally. That was a really cool talk. It's always neat to see kind of different strategies to approach stability over time and delivery and things like that. So thank you for that. And congratulations again on your poster award. So our next poster award winner is Sai Pradeep. And so I'll hand it over to you, let you introduce your affiliation and your role.
Yeah. Just let me start the.
Slides look good.
Hi, everybody. My name is Sai, and I'm a PhD student in Dr. Raman Bahal's lab at the University of Connecticut. Before I start, I just want to thank the organizers for giving us this opportunity to present our work at the conference as well as over here. This is a really nice opportunity for us. So to begin, my talk is going to be on targeting undruggable proteins using next-generation oncogene silencing. So it has been known that one of the hallmarks of cancer is sustained proliferation of cancer cells, and this is often achieved by the overexpression of oncogenes. Now, therapies that target the overexpressed oncogenes, along with the chemotherapeutic drugs, have helped in improving survival outcomes in cancer patients. Now, in the recent years, there have been a lot of developments specifically in targeting mRNA as well as protein. But when it comes to cancer, the main culprit in all of this is the mutations that occur in the genomic DNA. And in my project, we were trying to come up with an appropriate treatment which targets the genomic DNA and can help in aid cancer therapy. Okay, it's this one. What is that? Sorry, yeah. So the idea of targeting the genomic DNA is not new. There are a lot of small molecules that can target the genomic DNA, like cisplankton and cyclophosphamide, and they cross-link with the DNA. But however, they are very non-specific, and this can often lead to a lot of toxicity. Peter Derwin's group at Caltech had developed polyamides, and these bind the minor groove of the DNA, and they can recognize about 5 to 8 base pairs. They were tested in vitro, however, their efficacy in vivo was not evaluated as much. And then, of course, there are triplex forming oligonucleotides, which are sequence specific, which can help in getting rid of a lot of the toxicity which is related to non-specificity, which we see. However, triplex forming oligos bind only to homopurine regions in the DNA, and they often can have low binding affinity and specificity. So what are the main challenges that we have when it comes to targeting the chromosomal DNA? The first is that the DNA is condensed in its chromatin form, where it is wrapped around histone proteins. And this is the first challenge that we would encounter. We would have to have a reagent that can bind to the DNA, even though it is in this form. The second thing is the two barriers that the reagent has to cross, which is the first is the cytoplasmic membrane as well as the nuclear membrane, in order for us to see any kind of efficacy. So in order to achieve this, we plan on using synthetic nucleic acids, which are called peptide nucleic acids. Peptide nucleic acids, or PNAs, have been used by other groups like Peter Nielsen's group as well as David Corey's group, and they have been used as an anti-gene strategy. Over here, we wanted to build upon that research and see if we could use this in vivo and check its preclinical efficacy. So PNAs are nucleic acid analogs where the phosphodiester backbone is replaced by an aminoethyl glycine backbone, which is neutral in charge. The backbone is, this replacement also makes it resistant to enzymatic degradation. PNAs bind with complementary DNA via hydrogen bonds, and they follow Watson-Crick base pairing rules. Now, this is a regular PNA, and in this project, we explored the use of gamma peptide nucleic acids. So gamma PNAs have a hydroxy group which is present at the gamma position, which forms a chiral center. And this gives it a couple of advantages. The first is it does improve the solubility of gamma PNAs in comparison to the regular PNAs. In addition to that, it also pre-organizes into a right-handed helical conformation, and this helps in it helps in increasing the binding affinity with double-stranded DNA, and it is also able to invade double-stranded DNA. So for our nuclear delivery, we used something called a nuclear localization signal. This is a group of basic amino acids like lysine and arginine. We used which help in transporting macromolecules into the nucleus. In this project, we used a classical nuclear localization signal, which is derived from simian virus 40T antigen. And there have been studies that have shown that because of the basic nature of this, you do see an increase in cellular accumulation, and we also saw an increase in the tumor accumulation, as I will show you. And for the design of the PNA, we used a tail clamp design, which consists of a tail region that binds to the DNA using Watson-Crick base pairing, and a clamp region that binds using Hoogstein base pairing. So to summarize, we are using a gamma-modified peptide nucleic acid that has a nuclear localization signal peptide, conjugated it to help in delivery, and the design of this construct is a tail clamp design. Now, as proof of concept, we decided to target the c-Myc oncogene. c-Myc is mutated in 50% of cancers, and it undergoes a chromosomal translocation, specifically in Burkitt lymphoma, and is the driver of oncogenesis in that lymphoma. c-Myc is also considered undruggable target due to its protein structure, as it lacks binding pockets. It is also mainly localized in the nucleus, and the mRNA has a very short half-life of approximately 30 minutes. So in this project, what we tried to achieve was using a peptide nucleic acid with an NLS. We targeted the genomic DNA of the c-Myc oncogene, and we explored its transcription inhibition potential. So my workflow consisted of the synthesis of the PNA as well as quality control. We then followed this by biophysical studies. And then we moved on to cell culture studies, where we looked at the gene expression and the protein analysis of c-Myc as well as the cell viability. We explored this in multiple lymphoma cell lines as well as other cancer cell lines. And we tested this as a monotherapy as well as a dual therapy, where we treated it with the PNA as well as histone deacetylase inhibitors and CHOP treatment, which is the standard of care for lymphoma patients. And finally, we looked at in vivo studies where we checked the efficacy of these PNAs in 3 different mouse models. The first was U2932 cell-derived mouse xenograft mouse model. The second was a patient-derived xenograft mouse model. And the third was a transgenic mice model. Over here, we were looking at survival as well as safety analysis of the PNA alone and in combination with HDAC inhibitors as well as CHOP treatment. So to start off with, the c-Myc protein c-Myc gene consists of 3 exons, and it has 2 promoters. So we designed 2 PNAs that could bind upstream to promoter 1, which is PNA1 NLS, and PNA2 NLS bound upstream of promoter 2. PNA3 NLS was a scramble PNA of PNA2. PNA4 was targeting the mouse c-Myc gene. And PNA5 had the same sequence as PNA2, but in addition, it also had a TAMRA fluorophore, which helped in visualization. Now, the PNAs were synthesized using solid phase synthesis. They were purified using reverse phase HPLC, and we used MALDI for quality control. So the first thing we did was an in vitro binding assay. So over here, we synthesized double-strand DNA, incubated it with the respective PNAs at different molar ratios. We observed that both PNA1 and 2 formed a complex with the double-stranded DNA, which can be seen by the formation of the retarding band. However, the scrambled PNA did not show that. Next, we wanted to check how this worked in cell culture. So we treated U2932 and RASI cell lines with the PNA for 24 hours, after which we isolated the genomic DNA and then we amplified the region where the PNA would bind. And what we observed is when we treated the cells with PBS, which was just our control, or scrambled PNA, we observed the formation of the amplicon, which was not present when we treated it with the perfect match, which is PNA2. And this was consistent in the other cell line, RASI, as well. In addition to this, we also treated the PNA at different doses, and we observed a complete disappearance of the amplicon at an 8 micromolar concentration. In order to visualize the uptake, we used flow cytometry, where we increased the dose of the PNA in a dose-dependent manner and observed an increase in the cells that underwent uptake. We also used confocal microscopy, and we saw the overlap of the TAMRA PNA with the with the blue DAPI nucleus, and this confirmed the uptake of the PNA in the nucleus as well. And since this is targeting the genomic DNA, we did also some genotoxicity analysis, which over here we used two different assays. One was the gamma H2AX assay. And as you can see, when the cells were treated with the PNA, we do not see any gamma H2AX foci formation, whereas treatment with the positive control bleomycin showed the presence of these gamma H2AX foci, which was quantified. We also did a Western blot analysis and observed no difference in the gamma H2AX protein. And in addition to that, we used flow cytometry to just have a total count of the number of cells which would have the gamma H2AX foci, and we did not see any difference between PBS and PNA2. We also used the COMET assay, which is another sensitive assay to detect double-strand breaks, which can occur in the nucleus. And we did not observe any comet formation in PNA2 treated cells as well as PBS, but with bleomycin, you can see the formation of comets. Next, we moved on to cell culture, where we looked at efficacy. Over here, we first used U2932 and RASI cell lines, which are lymphoma cell lines. And as we increased the dose of the PNA, we saw a dose-dependent decrease in the cell viability as well. In terms of c-Myc mRNA expression, we saw that when we treated it with PNA2, we observed about a 50% reduction in both U2932 as well as RASI. And EZH2 is a downstream target of c-Myc, and we also see a reduction in EZH2. These results were consistent in our protein analysis as well, where we observed a 40% as well as a 50% reduction in the c-Myc protein levels in U2932 and RASI. So this was an interesting result. So we moved on to check how the PNAs fared in other cell lines, where such as cervical cell lines, hepatocellular carcinoma cell lines, triple-negative breast cancer cell line, as well as prostate cancer cell line. And as you can see, we approximately observed a 50% reduction in the c-Myc mRNA across all these cell lines. In terms of dose optimization, we did test different doses and we looked at the c-Myc mRNA expression. You can see that there is about a 50% reduction in c-Myc mRNA expression at 8 micromolar, but further increase of the PNA does not really show us more of a reduction. In order to check if our treatment was specific to the DNA, we also purchased ASOs that had the same sequence as the PNA. And when we treated ourselves with the ASOs, we observed that there was no reduction in the c-Myc mRNA, which would mean that the ASOs were not able to, that sequence was not present on the c-Myc mRNA, and hence we were not observing any reduction. And this result was the same as well in the protein analysis that we did. Now, since c-Myc is a transcription factor and it has an impact on a lot of different genes, we used RNA sequencing analysis to have a look at the transcriptome of these cells after we treated it with the PNA. We also used an siRNA control and we compared how the silencing effect was in both these groups. We did a longer incubation of the PNA and compared it with the control where we observed a total of 489 downregulated DGs, out of which we did see that we validated a lot of this using RT-PCR, and a lot of the genes that were downregulated were involved in DNA replication as well as repair. And downregulation of a lot of these genes can also help in sensitizing these cells to the chemotherapy drugs that we use. And that was something we also saw when we treated the cells with the CHOP treatment as well as in vivo. So the first combination treatment that we looked at was the use of histone deacetylase inhibitors, which are known as HDAC inhibitors. Now, DNA is usually bound around histone proteins, and the acetylation of these histone proteins can neutralize the positive charge on the histone, and hence it opens up the chromatin. So our hypothesis was that if we use HDAC inhibitors, that would help in improving the accessibility of our PNA to its target site. So we decided to use this for our combination treatment. Now, there are several HDAC inhibitors which have already been approved for lymphoma. And we have tested out a couple of them. Over here, I'll be showing the results mainly for the co-treatment of the PNA as well as romidepsin, which is a HDAC inhibitor approved for CTCL and PTCL lymphoma. So first in cell culture, we would treat the cells with romidepsin as well as the PNA. And what we observed is we observed a much greater downregulation of the c-Myc mRNA when we had the co-treatment, and this was significantly lower than what we observed with just the PNA alone or romidepsin treatment alone. And this also translated to a reduction in EZH2, which is a downstream target. And this was consistent for both the cell lines that we tested. And when we looked at the protein, the Western blot analysis, you can see that the c-Myc protein completely disappeared when we had a co-treatment of the HDAC inhibitor along with the PNA2. So now we moved on to our in vivo studies. Over here, we first looked at biodistribution. And we used systemic injections in cell-derived xenograft mouse to evaluate this, and we observed that the NLS did help in passive targeting of the tumor. And this was a little surprising because we did not expect it to happen. We did also do cryosectioning of the tumor, and we did see that you could see that the TAMRA PNA was present in these tumors at about 6 hours as well as at 24 hours. So the first thing we did was we started testing these in U2932 cell-derived xenograft mouse models, and over here we conducted a survival study. So when we would implant U2932 cells, and when the tumors gained a volume of about 100 to 150 mm³, we would start dosing them with the PNA, and our terminal endpoint was when the tumors reached a volume of 2000 mm³. What we observed that with the PNA, there was a retardation in the tumor growth, and this led to an increase in the survival of the mice, whereas the scramble control and the untreated were pretty much the same. Once the mice were sacked, we did immunohistochemistry staining, and we looked at Ki67, which is a proliferation marker. And you can see that the Ki67 staining is much lower in comparison to the scrambled PNA or saline. We also looked into caspase-3 levels, which is an apoptosis marker, and you can see that there is increased apoptosis in these tumors after the treatment. We did a short-term efficacy study in both U2932 and RASI xenograft mice, where we treated the mice with a high dose of the PNA, sacked them after 48 hours, and then looked at the protein levels of c-Myc. And over here, we observed about 50 and 40% reduction in c-Myc in the xenograft mice. We then tested this in combination with the HDAC inhibitor romidepsin, as well as with CHOP. And in both these treatments, you can see that the combination treatment of PNA as well as romidepsin or PNA and CHOP had a much slower growth in comparison to just romidepsin alone or CHOP treatment alone. And this resulted in an increase in the survival of the mice. We performed immunohistochemistry, and in both these treatment groups, we saw a decrease in the proliferation marker Ki67 and an increase in caspase-3 levels in these tumors. Next, we evaluated the efficacy of the gamma PNA in a PDX mouse model. So these tumors we got them from NCI, and they were a diffuse large B-cell lymphoma tumor, which we implanted. After which we treated it with romidepsin and the PNA, and we saw that the groups that received the combination of both had a much slower growth. And at day 35, you can see that even the tumor volume was much lower in comparison to the treatments used alone. Immunohistochemistry staining showed that the Ki67 marker was much reduced in the combination treatment in comparison to the other groups. We did the same study when using CHOP in the PDX mice, and we observed similar results where we saw a retardation in the tumor growth, a significant reduction in the tumor volume, as well as a reduction in the Ki67 proliferation marker. And finally, we looked at the efficacy of this in EUMIC mice. So EUMIC mice are transgenic mice that mimic the translocation of c-Myc that happens in Burkitt lymphoma. So basically, the c-Myc promote gene is under the control of the IgH regulatory element. And this results in an overexpression of c-Myc, which can lead to spontaneous lymphogenesis. So these mice just produce tumors spontaneously. So we first looked at two different dosing schemes, that is IV and sub-Q, and we saw with the sub-Q dosing, there was better accumulation of these constructs in the lymph nodes, which can be seen in the cryosectioning as well. So we did a short-term study over here where we just treated the mice, and then we would sack them after a particular period of time. And then we looked at Western blot analysis, where you can see that the protein levels are much lower in the treated group. Similarly, in the cervical lymph nodes, we saw that Ki67 was reduced as well as caspase was increased. And finally, since these mice have a proper immune system or an intact immune system, we looked at cytokine array analysis. We analyzed pro-inflammatory cytokines, and we did not see an increase in any of these in comparison to the saline-treated group, which showed that these constructs were relatively safe. We also conducted some other safety evaluation in C56BL6 mice with an intact immune system. Over here, we treated it with high doses of the PNA, following which we would sack the mice, and we looked at the immune cell profiling, where we looked at NK cells, T cells, B cells, granulocytes, as well as monocytes. We saw that there was no significant difference in these population levels between both groups. We also looked at monocyte uptake and saw that there was not much uptake in the monocytes, and we did a cytokine array analysis, and we did not observe an increase in any of the inflammatory or pro-inflammatory cytokines. So in conclusion, we had shown that the PNA targeting the P2 site in the c-Myc locus inhibits transcription. The NLS helps in delivery of the PNA into the cell as well as accumulation in the tumor. We showed that systemic administration inhibits the growth of the tumor in both the CDX and the PDX mouse model, and the combination treatments improve this efficacy. So in conclusion, I hope I have tried and convinced you about the use of gamma PNAs as an anti-gene agent. For our future directions, some of the things which we are exploring are, one is chemistry-based modifications, where we can change, we can use Janus base pairs that can bind to both strands of the DNA. The other thing which we have explored are combination treatments, where we have used the c-Myc, the anti-gene PNA with a host of direct and indirect inhibitors. And the third is we can always improve on the delivery, where we can use active targeted delivery to deliver these constructs to whichever organ or tumor which we would want to deliver it to. In conclusion, I would like to thank my advisor, Dr. Raman Bahal, for his guidance throughout this entire project. All of the work I presented here was mainly done by Shipra Malik and me, who's a co-first author on this paper. She is now an API. I would also like to thank all our lab members, our collaborators at Yale University, as well as all of our funding sources. These are some of the other papers that I have co-authored over my PhD. And yeah, thank you. And if you have any questions, you can always reach out to me. I've given the email address, which you can reach me at.
Great. Thanks, Sai. That's awesome.
Well, excellent presentation. I think this must be lots and lots of work. I can imagine that it took multiple years to get all these data all together. So my first question would be on the off-target effects. So how important is it to assess all the off-target effects, and how easy is it to assess these? Because I can imagine that it might, especially in these tumor models, it might be quite difficult.
Right. So the off-target effects, we did do a couple of things in order to assess it. We had the RNA sequencing data in which we could look at all the genes which were downregulated upon our treatment. And from the NCBI database, once we use our sequence and blast it against the human genome, we can find the other genes that could be impacted or have any sequence similarity. And what we observed is between the downregulated genes that we observed from the RNA-seq versus what we got from the blast analysis, we did not see much of an overlap. There was no overlap. So that was one way in which we assessed the potential off targets. However, in order to be more comprehensive, I do think you would have to do like a whole genome sequencing. So that would give you like the most well-rounded answer for that. Yeah.
Yeah, but so also I think when you see some effects on genes that you might not, so it's not necessarily the on-target off-target effect, but it can also be the a secondary effect of an off-target effect. So it's always difficult to make the discrimination between on and off-target effects. Um, One other question that I have, because I think you mentioned it already on your last slide. So how to improve the delivery in the end?
Yeah.
Because that I think it's still one of the main challenges.
Yeah, so over here we used NLS, and what we did was, if I could just. So this was a preliminary study that we had used. So this is the PNA with the NLS, and over here, this is the PNA without the NLS. So you can see that with the NLS, you have more accumulation of the PNA in the tumor. So we did observe like a passive accumulation, which could be due to the EPR effect. But in case we want to do a more active targeting, then it's always better to have like some other peptide conjugated to it. One peptide that has shown to be effective in targeting tumors is the flip peptide. And of course, if you want to target other organs or anything, you could always use some ligand conjugates like GalNAc or lactobionic acid. So that would be more specific to an organ. Yeah.
Yeah. So a question from the chat. So you use the nuclear localization signal. But are there any other ways for to direct the PNAs to the nucleus rather than using these NLS?
NLS peptides are the ones which I have read most about when it comes to even other groups that have tried to target either the nuclear DNA. NLS sequence, I mean, there are a lot of other NLS sequences that you can explore because we used a classical nuclear localization signal, but there are non-classical nuclear localization signals which people have also used. But apart from that, you could also try conjugating it to a small molecule, which would help in transporting it to the nucleus. So yeah, I think there are other ways that could be done, but over here we are sure that the NLS sequence that we are using interacts with important alpha, and we have translocation of the construct into the nucleus. So it's a very well-known transport mechanism. So it was the best bet.
Good. Going back to Aaron. Not sure, Aaron, whether you have a last question or you want to close the webinar.
Great. Yeah, I think for sake of time, we'll close. But I just want to thank Sally and Sai again for excellent talks. Congratulations again on your poster prizes. And I just want to remind everyone that we have one more webinar coming up before the end of the year. It's on December 11th. So please make sure you register and you tune in. It should be a good one. And again, if you have questions, please reach out to the speakers. They provided their contact information. And you can also email the info@oligotherapeutics.org.